So over to you, sir.
Okay, thank you, Dr. Vikas. Yeah, thank you. Yeah, just confirm my screen is visible.
Yes, sir. It is visible, sir.
Yeah, okay. Thank you, everyone, and welcome back again. I hope you refreshed yourself. This one is on brain-inspired networks in a way that we uh, we try to model the networks which are more near to the biological brain, and they are known as basically this category. They are nomenclatured as spiky neural network. So that is SNN. So in this during this discussion, what we will do is we'll try to start with why we need it, and then how the things are actually biologically happen. So we move that one, and then how those things can be transformed into uh, some kind of mathematical formulation, and then towards a spiking neural network. So that's the workflow here. Uh, let's start with a domain, and that is the neuromorphology. So there are three parts here. Neuro is the nerve system, morpheme is form, and logium is the study of it. So that's how the neuromorphology and people study it in various aspects. So three main domains are there. So the nervous system, we have synapses. Try to model the synapses in a way, and that is basically the neurons. So neural network which we are using, one form, this one is coming here uh, as a neurons, complete neuron. There is a some form, another form, which is synapses, basically at the end of the neurons, how one neuron is connected to the another neuron. So people are working at that level. The another level is basically the transmitters. So we know that these neurons, they transmit some chemicals, and another neurons, they bind to it. So how those transmitters and the binding process and all those occurs, that is a one part of it. The another part is of neuromorphology, is what is the structure of the brain, and then how the memories and all those things are developed, or the zones they work. The another domain is basically the networks. So how the neurons, they form a network inside it, and what is the cognitive process, and then another area is that what are the things which are responsible for neurological disorders. All this comes under the neuromorphology, and the researchers, mathematicians, computer scientists, those are working in the domain of AI. They are using uh, these sectors to develop a certain kind of a mathematical model, just to mimic the how the things are happening in the real brain kind of brain, and the connected nervous system. So we know that we have nervous system and categorized into two main parts, that is CNS, that is central nervous system, and peripheral nervous systems. Both these nervous systems, they have different job there, uh, but the main function of the system is it collect the sensory information, and then it do kind of integrations, and then send it to a certain kind of processing domain, and where the response is taken back to the motor outputs. So you can, and the processing and all that is done, and the brain, where we have cerebellum, and the spinal cord helps to taking the signals to the through, I mean, through nervous system, take the signals to that process part there. Not going to the biology of more biology, but we can see some biological part in the coming slides there. And then the neurons, basically, you can see the structure is like this here. The structure is there. You can see the information is coming from other parts, those, and they are connected to the dendrites. So dendrites take a signal, the signal is in a way summed up here, and then the signal is transmitted through this action part, and then the last part is again connected to the next neuron, and the connection point junction point is like this, so which is known as a synapses. And you can see these are the yellow points which contains the transmitters, so they transmit the another part, they bind with the other part, that is the receptors, and how the signal is transmitted. So the whole flow is something like, okay, we receive some signals from mother, and then the signal summed up, and then the signal keep on moving, and then they reach to the endpoint and connected to the next one, and this is how the process occurs. uh, based upon this working, the first type of so we can I'll come to that part, but we can see that how the things are happening, and this is already we have discussed in a way that these neurons are interconnected with each other, which uh, conceptually been taken to develop a neural networks, that okay, how they are connected with each other, and the connection is always weighted one, so we have weights there, and then one segment of it is like this, so this is basically the neuron which we are talking about. So we are always we consider a single neuron as like, okay, it is taking some signals, so there is a inputs, and if it is a network, then there is an input layer, and there are layers in between, we call as an hidden layers, and then the third mechanism is how we learn it, that is basically adjusting these weights, and that is how all this occurs there uh, in in our neural network, as well as a bit in a biological neural network. Okay, so the point here is initially it comes like this. So initially I told you that okay, signals are coming and they are reaching here at the dendrites, and they are summed up, and then they keep on moving to this path, and at the end you can see this synapses, and then they are transmitted, and then the signal is transmitted to the next part. But you can see that uh, yes, Dr. Kanji Moses, you have any question? You have raised your hand. Is there any question right now? Is there any question right now? Okay, okay, then the question will take later on. Okay, so the initial model, people take it like this, that signals are coming. Okay, these are weighted signals. What is the weight? And weight is basically when the signal is coming from another neuron. It's not like the 100% the signal is transmitted. Some part of the signal is lost also. So that is basically, and the another way is the amplitude of the signals which is coming from different neurons, that is different. So we can take it that as a in a way like, okay, weighted kind of a signal which is reaching here, basically the central part, and then that one is summed up, okay, and which is done here, and then at the end you'll find there is a gap here between these two neurons, and if the value is sufficient enough, the value of the signal which is here is sufficient enough to fire these transmitters, only in that case that transmitters, they or excite these transmitters, they move and they go to the next one, otherwise not. So that they in the first generation they mimic it by this function, step function. If the value of the sum of the signal is more than this one, then only the signal is forwarded, as not forwarded, and that is basically the first generation of the neuron there. And we know that we recognize this one as a perceptron. Then later on, there are issues. People find that this is more complex. This fails to solve more complex problems, and then there are issues with this function also, because this is a discontinuous function, and then we need a more non-linear mapping. This is hard linear. So then people go shift from this to the second generation of neurons, where they instead of taking this hard one, they have taken some kind of a function which is a continuous and which is bounded also, so that we have better results. We have non-linear mapping and it's a bounded weighted solution, so that we can reach to it's not like a weight explosion kind of setups there. Well, after what happened in these two scenarios, and that is the important part here. So we have artificial neural network, and we know that the process is sequential, okay, one by one, and it's centralized. Control mechanism is basically we can put it external as well as internal. Complexity, it can form a simple problems, but if the problem become more complex, we have to increase the size both initially in the initial stages. If you see the literature, people make fatter neural networks, but now they are going for a deeper or longer neural network, which is a deep learning kind of a setup there. Reliability is still a challenge, and the energy use is very high about these kind of arrangements there. You can take that only a simple thing here, that like OpenAI and ChatGPT 4 and that Metalima, they 3.1, they consumes approximately 30 megawatt during training. So the consumption of power is very high, and reason is obvious. In the ANN which we have studied so far, there is a continuous input is continuous, which you want to realize practically. It consumes a lot of power. Continuous input means that input is always connected to a power source, which is not actually in the brain. And if you want to make a real hardware on that one, then whatever you develop, that contains a lot of, that consumes a lot of power. And that is basically one kind of a thought people excited that to the researchers also, like, can we develop a more kind, because the consumption of power in human brain is very, very less as compared to these machines. So can we go more nearer to the biological neuron and develop a neuron like that, so that we can have a low consumption and high accurate and better accuracy models for estimation, and that's how these things comes into the picture. Okay, so you can compare it both biological and here. So one is the parallel, and then it's a robust biological, and then the important part is which initially excite many of the researchers, that is it consumes very less power. So then how to do it, and likewise. So there, that is the third generation comes in, where the input which is coming, it's not a value, constant value, but it's a train of spikes basically. And we know that the spike is there is an amplitude, but the amplitude time is very, very, very small, and then it becomes zero. Then it's a then the spike, and then it's zero. So if you consume the power requirement, that is very less if you compare it with the constant one. And and that is our today's agenda, that how the spiking neural network works, what is the philosophy, what is the biological aspect behind it, and then how uh, mathematicians convert it into model form, and then how we can use it for our purpose. So their characteristics are spatio-temporal, asynchronous, sparsity, and energy efficient. These are the characteristics of this one. And there are some components of spiking neural network. Those are encoding schemes, because we need to convert a real number into some kind of spike trains. So what scheme we can use, and then the model and the learning also. We'll come to these things one by one, but before going to that one, let's understand about the uh, neuron models and what we can take it from here. This one is a typical neuron model which are there. We can do few things. We can develop a neural model. So now we are talking about developing the neuron model, conductance based. So this is how, if you go into the models of the neurons, they are characterized, or the development is on the basis of few things. One is conductance based. When we talk about the conductance based neuron models, we talk about ion channels, how ions are how the ion channels are working. So ion channels are there in our membrane. I will discuss more on that. It's a biological process. And then how ions move, what is the membrane potential, and what is the dendrite morphology, and what are the biophysical properties of the neuron. So these are the areas when people have explored which are there in actual biological neuron to develop certain kind of model. There are other models which are phenomenological models where we talk about input-output relations and then spike generation, time, constant, noise. That is the behavior part of it. So in this conductance based is normally very popular in the computational neuroscience to study neurons, memory, and related diseases. Whereas the behavioral part, the another part that is phenomenological part, which is more popular towards deep learning, improve the plausibility and efficiency of these models there. Okay, so I've taken few slides and you can check it. Entrim Karisnov channel also has presented a very nice animations there to understand the biological process there. Okay, so we have a cell membrane. So you have this one is the cell membrane, and then it look like this. Okay, and then what is there on the both sides of this membrane? We have ions, and we have both type of ions. We have potassium ions, we have calcium ions, and we have sodium ions. And you can see from your knowledge of chemistry, they have we know that potassium and sodium ions are positive charged ions and the calcium ions are negative charged ions. And you can see both the ions are both the sides, but the concentration is different. You can see the potassium, so your sodium ions are more outside than the inside, and you can see the potassium at this stage, and you can see the potassium ions are more there on left hand side than the right hand side. Well, this kind of structure, and this is actually there in the brain. So this kind of structure, this kind of arrangement is the basic of that how this spikes and all these things work. So we will try to connect it happening there with certain kind of understanding of chemistry as well as electrical properties here. So we know that if the ions are distributed randomly, it's okay. They will not affect that much because they neutralize each other. And we know that if we put a membrane there, and if the membrane separate the charges, say that positive on one side and negative on another side, then it will develop a potential. It will develop a potential difference between both the sides, because on one side we have positive, on the another side is negative. If it is not allowing anything to pass across it. So if it is not allowing anything pass across it, it will develop a kind of a potential difference, and which is a voltage there. Okay, and if from the basic property of physics we know that this voltage is basically, we can correlate it that this is equivalent to a capacitance. So the charge of the capacitance can be mathematically described with the capacitance value into the voltage. So in total we can say, okay, voltage is developed across the both sides of the membrane there. Okay, and we know that the rate of change of charge can be considered mathematically, can be described assuming the capacitor properties are same as the rate of change of the voltage. And we know that what is the rate of change of charge is that is a current. So that's a basically electrical way of defining the things, but it is not there. It is not like they are not permissible. This equation holds true if we see that nothing is allowed to move from this side to this side, or likewise. But these membranes, they are semi permissible. That means they allow ions to move across the membrane, and they allow through certain gates. So you can say there are certain gates here, blue and red, and rest of the membrane is a solid, it will not allow. So only the ions are allowed to move across these channels, and these channels they open and close. That's the complex structuring, and let's understand it how that is happening here. So we have charges on the both the sides, and we have gates, and then when the gates open, they allow the charges to move from one direction to the other direction. So now my equivalent circuit of all this process is like this. If these ions are, if these gates are not there, so we have a capacitance who has a positive on one side and negative on the other side. That is means a voltage is stored in it. But if I have ions, I put two ions here, okay. So if they allow, then they allow to move, allow the movement of these charges, basically allow the movement of these positive and negative ions. Okay. Okay. So the current, we know that what is the current from the first equation, and then these ions, because the current flow through these ions. So that means there is a current flow. Current flow these two ions means, because we know that if the charge flow, then that is equivalent to a current is there, electric current is there. So that means the charge flow through the red one, the charge flow to the blue one, the sum of these charges that has to be equal to this one, this voltage here. So we have a very basic equation here, that the change of voltage with time is equal to the sum of the charges. This is an electrical circuitry property, and this exists in our brain also, that that this movement, this charge, because of these gates, if these are not there, then there is nothing, then the charge remains same at the plates. But since the gates are there, then that means they allow the charges to move. Okay. And this is basically a action potential. That means the how the membrane voltage occurs. So it will take certain kind of shape, some value here, and then it will increase it. This is a negative value, okay, and then it increases in here, means it become more positive, and after this point it goes back. But the point is that how this voltage is taking this spike, and this one is basically a spike. It look like a spike, and that's why this whole thing comes to it, that spiky neural network. Let's check it. So what will happen actually? So initially the voltage is very low, nothing there, but then sodium ion, basically sodium channel, this one, this opens basically. Okay. So once this open, because at certain time, if it opens, so what will happen? This voltage, say somehow it increases. We'll see how it increases. So this, at this value, this gate opens. Okay. So this will allow the more positive positive ions to move inside the inside the cell, and then it increases the potential, because positive appears here, more positive gathers here, so this potential keep on increasing. So when sodium ions opens, the outer sodium ions, they comes into it, and the potential increases. Once it goes up at a certain point, then it closes. So once it closes, then this part is closed here. So that means no charges they are coming, no sodium ions are coming to it, and then the potassium ion open. So then when potassium ion open, it open at this place. Once it open, what it will do, it will allow only potassium ion to go out. It will not allow sodium ion to go out that way. It will allow potassium ions to go out, and potassium is also positive. When it goes out of it, then the voltage inside drops. So that's why you will find this graph goes down. So you can, if you can correlate it here, what will happen is basically here at this instant, sodium gate open, the inside the membrane voltage inside it increases. At certain point it closes, and then potassium opens, and then potassium moves away, and so it goes down, and it's stabilized like this. So this is actually the formation of this spike totally depends upon the opening of these two gates. As of now, we'll consider rest of the things later on. Opening of these potassium and your sodium ions. But the question is how these, why these open or the voltage and all this occurs. So there are two interesting things which are occurring. One is this process, that is basically diffusion process. The diffusion process means if the concentration is more here, if the concentration is less here, then there is some force which push them to move from this side to this side. Okay, so basically statistically you can say that particles from high concentration, they are going for to the low concentration. And then the second operation which is working, that is basically electrical forces. So we know that like like charges, they repel each other, and unlike charges, they attract each other. That is the electrical force. So there are two types of forces they are occurring. One is your diffusion force, and another one electric force, and this help in that movement kind of there. So we know what is the electrical force there and all that. Okay, so if we talk about this one, so here you can see that there are negative ions there, it's not there, so there is a force which push this negative to other side because of a diffusion, and because this is positive, this is negative, there is an electrical force which attracts this negative to this side, and here this positive, they push positive to go to the other side. So that is actually what is happening here, and this thing is interesting here. So that and these two process, they depend upon two different things. Like diffusion depends upon the concentration. The concentration is more and the other side it is less, then it forced to go into that direction. And the electrical force, that depends upon this voltage, that what is this membrane voltage. Okay, and then this balance basically check it here, because that is, you know, that for this scenario, diffusion force push this potassium ion to move into this direction because the concentration is more here, it is less here, whereas the electrical positive outside, negative here, push it to go back into this. And depending upon which one is more uh, uh, and which one is less, the movement takes place. At equilibrium, these both forces are balanced, they are balanced with each other. Okay, so usually if we talk about say potassium ions, this ratio K in on K out mm, uh, around 30, and it will generate around at a potential, of 90 millivolt. I told you in the spike, the starting point, it's a negative voltage, so at 90 millivolt, and this is the ratio there. So if V is more than this, have a graph like this, a decaying graph, uh, because of the two types of the forces acting, which are acting there, and if it is other way around, then the graph is the other way around. Anyway, uh, so the current is now current is working on the two things there. And what are those two things? One is this one, okay, that is basically ion, which are the ion concentration, the voltage because of the ion concentration. And another one is your the voltage membrane potential. If they are balanced, then there is no any current because the difference is zero. But if they are not balanced, if anything is more, then the current will flow through them. That is a simple logic here. So mathematically we can write it, the current is proportional to this difference, and then we can take it is multiplied by a coefficient. So the current is equal to G, and G is a coefficient into the difference between the two potentials. G is basically a it's analogous to a kind of a conductance. So if there is a conductance, because we know that conductance is a property that how the current is flowing through it. It controls that current. Okay, so it is equivalent to your Ohm's law. So the current is their conductance, and we have voltage, so we can have I is equal to V by R. This is the Ohm's law, and we can represent G as inverse of R. That is basically the structure there. Now this G is basically it. Now this G is not as simple as anything, like as simple like, okay, we have a conductance, a constant value. No, this G is variable because it depends, G depends upon the gate open and close. And there's not a single gate, there are many gates there. Okay, so it's no more like in here in Ohm's law, we assume, we know that resistance is a constant property, and then we can say V is proportional to R, I. But here, this one is variable, and that makes this thing complex. So how people model it, they say that, let's say that I is G into P and the potential difference. This G is the maximum conductance when all the gates are open. So that is constant basically. Now it's constant. But now it is multiplied by P, because I told you here that this G depends upon how many gates are open and how many are closed. So if all gates are open, that is the maximum current that can flow. But if 50 are open, 50 are closed, that means only 50% contribution is there. And likewise, so they modeled it in a way that this G is the maximum value of the conductance when everything is open, and then this is multiplied by a factor which is a fraction of the open gates. We call it a probability. Okay, so how many probability, what is the probability of gates which are open? So that is basically P. Okay, so now this since uh, this is now variable, this product is now variable, and that makes our equation a reasonable one. But now the open, it's not so simple that, okay, we consider this P as a simple probability of this one, how many are open and how many are closed. It depends upon the complex protein mechanism which regulate this flow, which regulate the opening and closing of your gates. Okay, so how it regulates basically, so there are two types of gates, permissive gates and non-permissive gates. Okay, so you can see here in this picture, you can see there are four gates are there. These are four. So this one is the path through which ion can go. Okay, so if all the gates are permissive, then only channel is open. So it's like a lock and key kind of a system. When everything matches, only in that case your gate will be open. Okay, so there are two states, permissive and non-permissive kind of structure there, and then the mathematics, try to find out a mathematics for it. Okay, so they say that let n be the probability of finding a single gate in the permissive state. So they put it like dn by dt is alpha. Alpha is rate of moving to permissive state into 1 minus n, minus beta is basically non-permissive. That means it's not same, like moving from this to this side, non-permissive to permissive rate and permissive to non-permissive rate is not uniform. It depends upon there are two different rates which are known as alpha and beta. So this will give us the probability, because we want a probability that what is the probability of getting the ions open and closed, because at the end that will give us the current which is required for finding the potential and the spike train there. Okay, so if you try to plot these things, they depends on the voltage. So people try to find out the empirical relations, and then they find that, okay, a rate constant and the membrane voltage, so it depends on the membrane voltage. So this is the relation. So now this is now the more complex. Okay, just try to understand. My current equation depends on the conductance. Conductance value depends upon the probability of the gates. Probability of the gates depends upon the permissive and non-permissive, which is basically a differential equation. And then and it depends upon alpha and beta. Alpha and beta are the rates at which the gates open and closes. Okay, and then these alpha and beta, they are a function of the voltage. These are the function of the voltages. Okay, so all are coupled with each other. this relation was very well established in 1932 experimentally also. You can see the curves. These are alpha and beta curves. So they have done it. They measured the resonant membrane curves and current nerves, and then try to find out this relation. Well, I have uh, this relation for 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 one type of structure there. Okay, but this is only for one gate. So n is the probability for one gate, and there are four gates we have seen there. So for a potassium, there are four gates. So the potassium structure will be like this. We multiplied n to the power 4 here, because we need everything open. The probability of this into probability of this into this into this, everything open. Then only the channel is open. So the current of the potassium is a constant value, that's the maximum value, into n to the power 4 into the potential difference. Similarly, we can have this one is the potassium one. So we have all these equations now on single slide. That is this is the current equation, this is the probability equations, and then alpha beta, their dependency on the voltage. This is a potassium channel. Similarly, we have sodium channel also. So for sodium channel, now sodium channel has two interesting things. It has four, but three are of one type and one is another type. So there are two types of gate: activating gates and inactivating gates. But to open the whole structure, means to open the gate, all these has to be arranged in that manner. So the multiplier here is not n to the power 4, the multiplier factor is m to the power 3 and h. So h is basically inactivating gate, and m to the power m is basically activating gate. And same kind of equations you will get it here, but now the coefficients are different. We have coefficients for beta m and we have beta h. Now you can compare both the two. These are generalized equations. So generalized equation is current is nothing but G, which is a function of voltage, and then potential difference. Potential means we have constant term to the power multiplied by the n to the power 4, that is the fraction of the gates in the permissive state. And the sodium one is basically M3H. M is, there are two types of gates, and then what is the fraction of that each of being in a permissive state. This is basically for potassium and sodium ions. So this is the common equation. Okay, so I'm just going a bit fast here. And then there is current because of the calcium, which we put it like that, we call it a leakage current. So now my total current is sodium current plus potassium current plus leakage current. Leakage current is because of the calcium movement there. Okay, so this one is at one stage, and we know that our neuron is basically, we have axon and then we have sheath there, and there are multiple connections there. So this one is, the structure is placed like this. This is known as Hodgkin-Huxley equation or model, because the arrangement is like a cable. Okay, and if you write down the mathematical formulation of that cable, you can see all the equations are coming here. You can see it's almost the same kind of equations. You can see M3H because of the sodium, we have potassium, we have leakage, and then we have capacitance dynamics that is equal to the voltage across whole of the space. These are coupled equations. We need certain kind of mathematics to solve these coupled equations. That is actually how we model at a neuron level what is actually happening there. There are various models. So what we have discussed is basically that is Hodgkin's models, and the flops are 1200, and then people develop many more. So one of the popular, which is now very commonly used because it's easier, so that is LI model, okay, which has only 5 flops there. That is known as leaky, integrate, and fire model. Okay, so this one is right now very popular because it match with your experimental data. Computation is easy, integration behavior is also there, and implementation because of less is easy because of less complexity. So this one is what is leaky and fire model. Let's move to this part. So I have one input which these pulses, and then I have another input which these pulses, and then I sum them up, and then they pass through the threshold, and then they generate output. So you can see everything is generated in the form of these pulses there. Well, if you are looking from the biological aspect and going to there, you need all those calculations which are you need to understand how this one is happening there. But for a computer science person, it is not that important. For a computer science person, important is how to get these spikes there. Try to understand this part. The earlier part is when we try to work in computational neuroscience. But if you are working on the AI and computing part, that part you just can understand in a way that okay, a spike is generated. But then the next thing is how we use that spike for our computation process. So there are ways to do it, and that is known as neural encoding scheme. So now we are discussing more about what is actually happening there. There are 4 schemes which are very popular. We don't need that complex mathematical equation solved. We need a mechanism to represent our numbers or pictures or values in terms of some spikes. So how to convert those things into spike? So there are 4 schemes which are very popular. You can see these references which you can take it. One is rate encoding mechanism, another one is temporal encoding, another one is population encoding, and another one is the sparse encoding. So what is the rate encoding scheme? Rate encoding is basically what we can do is we can fix a rate, okay, that okay, this is the rate firing rate, and then if I know the firing rate, then what we can do is we can convert the signal value to that particular rate kind of stuff there. Like you can see here, you can see that during this period, we divide this whole period into 6 time steps. So during every time step, there is only one spike. Okay, so that is a basically more representation of the frequency of the. So this is equivalent to a value 1. If I look for value 3, that means there are 3 spikes in every sample. If I am looking for value 2, there are 2 spikes in every sample. That is basically rate encoding scheme. So the algorithm is very simple. You define the total time of window of your encoding. You fix a maximum rate, get the input signal, and then find out the define a rate function. There are various rate functions available. Somebody use Poisson's process, or somebody has do something, and then you just generate a spike from there. It's a representation of that one. The second one is much simpler. That is temporal encoding. So temporal encoding is basically through the timing of the spike. Okay, so say if this is the process of, so here this signal, if I want to take a second value as a 2, the representation is the firing will occur at in the second segment. If it occurs in the 6th segment, the value will be 6. If the spike occurs in 4th segment, the value will be 4. That means at the value is converted at what time the spike is occurring. So again, the process is very simple. You define the total time, then define the segments there, and then according to the value, you generate the spike. That means that is basically a temporal encoding kind of scheme there. Then another one is the population-based encoding scheme. So we divide this whole segment into some kind of functions there, and then depending upon the value, we try to see that how many parts it is affecting. So it is giving the population here. So like a value, some value somewhere here, so it will give me the spikes. It will cut various lines at various points and then give a concentration. So this number of spikes at these time differences or degrees, we can generate it and we can use it. We can use even a multiplier also for this purpose. There are various ways to get it done this way. And then that is a sparse one, that like, okay, cat, I'll represent it like this. So if I put it in a line. So that means it can be, okay, third neuron, it is 1, and then all 0 0 0 0 0 0 0, and then again 1, and then all 0 0 0 0 again 1, and then all 0 0 again 1, and then we'll say that, okay, this is a representation of a cat. So it's a sparse activation calculation. So if neuron is fired, we take it to 1. If neuron is not fired, we take it to 0. This is another way of converting your real value to a sparse encoding values there. That is the first step. The second step is how you make your things learn. Okay, and learn means, because we need weights, otherwise how the network will learn. So we know how to convert my inputs, real values, to kind of a spikes there, but the next part is how to train it. So as we discussed in earlier part, if we have this part neuron like this structure, kind of this thing, we take a derivative, it will create a problem there. What we do is we go for approximation kind of stuff. We make it a surrogate function, and then we use a back propagation to do it. That is one way of doing it. And the second one, this one is known as surrogate back propagation technique. The second one is known as spike time dependency plasticity. This is more towards a brain, because we know our brain has a plasticity. So we look for two things here. We have two curves here. One curve represents positive and negative in a point. It's a potentiation and depression, and that occurs in the brain. In brain occurs, we both have potentiation and depression, and we know that this curve is a smooth curve, so we can use this one for our purpose to learn the algorithm there. Okay, so surrogate backpropagation is similar. You have loss function, and then you can use it, the derivative, and to solve the problem. So, okay, we use a surrogate function, which is a bit a kind of a continuous function, and then we can use the derivative properties to train our models. That is basically how we use the surrogacy here and to train our network there. Okay, in spike time dependent plasticity, this one is basically more towards unsupervised way of process. So rules are based on the local information, and there are two parts here: long-term potentiation (LTP) and short-term depreciation, because this is not a very symmetrical one. It is a bit asymmetrical curve there, and we define it in terms of mathematics. So delta W, we can take e to the power some time constant. So you can see here what is happening here. Now the process is a bit different here, and delta t, it actually works on the process like what is the response of pre and post. That means at spiking trend, what is pre to it and what is post to it. Though both the time periods are noted here, and based upon that, this structure is being developed and the weight is considered accordingly. So the choice of the exponential function depends upon you. Here this equation comes through experimental data, because later on we have also proved it, how the concentration of the neuron changes during the learning, and then we proved it that is in line with the biological concentrations. So that can be this can be these equations can be proved, but with certain kind of restrictions. They are very much near to the human brain processing there. So how it occurs, you can see here we have pre-synaptics, we have post-synaptics, and then how much is the, from that time of occurrence, we try to find out this delta W. So we know that pre-synaptic train, what is my pre-synaptic spiky train, what is my post-synaptic spiky train, and then from there we find out this delta T. Tau is the time constant, and then A is a learning rate, and based upon that we find out the learning factor, and we update our weights. So it's not like a gradient based and all that. It is more on what is happening pre-synaptic and post-synaptic. So what are the change in the time period of the change in the, not time period, change in the occurrence of the spikes. between pre and post. This is more of a more near to the biological process. So this is another way to update your weights. Okay, so this one is a simple explanation that long-term potentiation and depression, because one curve is going up, another is going down, and then how you can calculate it. This is just a mathematical representation that we have time of presynaptic. This is the pulse. Postsynaptic. This is the pulse. We find out what is the difference. These are our learning rates. We put it here. We this one is our time constant of that particular neuron, positive and negative side. So from there we can get the change in the weight, and then we increase or decrease the weight depending upon the situation, how the spikes are being generated. So it's a derivative free kind of approach here. Okay, so I'm just skipping this part. The next part is how there are different ways. One way of using SNN. One way of using SNN is you do it everything from the part 0. There is another way that you can convert your ANN model to SNN model. Okay, conversion techniques, we can use the same like rate based. There are 3 popular conversion techniques. That is rate based conversion, temporal coding, and weight and threshold balancing conversion. This tool is available SNN toolbox, where you can use this thing. So basically you can convert your ANN to SN, and then you can use it. So how it is occurring? Let's take 3 cases. So rate based conversion, you can check it. What are the steps? Rate based conversion, it maps activation values. So there is the whole process is occurring. So the activation of ANN is converted into spiky rate. That's all. So what is the input? Input to the model is ANN, trained ANN weights, activations, and time steps. And then we fix the maximum rate. And after that, based upon that, the rate, using this rate, we convert those things into the spikes there. So the rate based, why we call it the rate based conversion? Because the conversion is based upon that what we have discussed, the rate encoding kind of a technique there. There is a temporal coding conversion. So it converts your activation to spike latencies. Okay, so if the higher activation leads to earlier spikes, so it's a time setups there basically. High value, it come earlier, low values, it come very late, and some kind of disarrangement. These are the well established and used in the research articles. And then there is a weight and threshold balancing kind of this thing. So you can see the difference here. So these values, they are converted into the spikes. Now these conversion to spikes depends upon the approach you are taking it. You are free to choose any approach. We have weights, they are summed up. So actually this is not a now how you are summing it up, that is basically the interesting part. So this one is actually the representation like a single spike, but it is not this one. This one is a some time constant and the spike is there. So when you sum them up, it comes like this, and then when it passes the threshold, it will generate a pulse there. So how you are summing up, that is again there are certain articles you will find that how you are summing them up, and then how you are generating a spike from the threshold. So the conversion is here, the conversion is here also to get the things done, and you can have the very interesting some kind of arrangement here also. You can see the weights have they are modified, but you can see they are labeled with a certain kind of potentiation and depression also. So those things you can add to your structures depending upon the complexity of the model, or the model you want to develop, how much near to the real biological neuron you want to develop it. So those things you can take it from here. Yeah, okay. So I think I am able to finish in time. I kept it 5 minutes for the questions. I can see there are certain questions. If you have more, you can type it. Uh, there are certain things. Okay, I can see one question is general question for session and uh, a general question for session and considering the problem of low energy usage, we want to mimic organics electrical signals. This is not only for DL but also for efficient data communication, which are some progress. What are the some progress in this instant computation and data transfer done by synthetic tissue and for see yeah. So what I discussed with you is basically try to connect the biological mathematical modeling of the neuroscience in a way and then connecting into electrical signals because we know that the that there are these are the signals which are moving, and then people have done the experiments and they find out the the currents and the charge of the membrane and all those things there. Energy is always an interesting feature, and not only as of today, long back people when started about uh, you can talk about Hopfield, and from that time till today, energy is energy models are there, and people are using energy models to develop the solutions. And we know that always we look for a lowest energy so that the system stabilizes there and all that. And you can combine your energy models here in for SNN also. But the only thing you need to see it where you are combining and for what purpose you are combining your energy models. And we know that lower energy models, okay, the data movement and all those things are they are working on it. But the agenda what I have taken here is just to make it a clarification is to connect the biological happening with the electrical happening and then to connect it with the SNN model. There are various ways to present your SNN model. I hope I'm able to uh, answer your question there. Because as such a computer or AI expert, which one Preeti or question is it in production? What in production? The energy models? Yeah. So SNN practically people have developed some chips which we are also trying at the chip level designing a chip which can be used which can consider as equivalent to SNN model. Yes, people have developed chips to SNN model, and the good part is because we have also proved it in one of the article, the energy requirement is very low. So if we are looking for a hardware, these can be a very good potential for the future because they consume very low energy. Just you need give a spike, and then rest of the things will occur. Then for some time, the system is idle. So that makes it more uh, one can say energy efficient. Yeah. So and people are working more on in this domain also. Uh, NN to SNN conversion, yes, people are using. So what are the pictures I have put it there, the flow charts, they are taken from reference articles. I forget to put the references there. Apology for that, but you can search it. Those are the published articles, how to convert from so ANN to SNN, and people are now combining various things there that they are using SNN at outer layers or they are using SNN at inner layers and they're trying to figure it out where is the more consumption, can we replace that section by SNN or can we replace the whole of the section with SNN. Those research are uh, uh, those research areas are going on. Yeah. Yeah. If you are interested in computational biology, then there are lectures, there are very good lectures, talks from Toronto. You just search it from the Toronto University. There are very good lectures. They talk from very biological aspect, and then how to model into the mathematical formation. But there are few lectures like one part I have shared with you. So I think one more lecture of his is you can find it on YouTube. Is very interesting for you to see that how the neural dynamics come into the picture. So he has only few lectures, but the animations and the explanation part is done very nicely by that person. And then on SNN there are books. There are very good books which you can refer. Right now I'm not able to recall it. I'm working more on the biological side of it, but I'll see if I can if I can search and share with the Dr. Vikas or Dr. Surender during this course time. You can download it from there. Any questions? Any more questions if you have?
So I think there is no more questions, sir. And the session was very well ordered and nicely explained, sir. I was asking Surendra answer the same question the Preeti ma'am has asked for any
Uh-huh.
further references, because
Uh-huh.
we are not getting that much resources for this particular topic. So
Yeah.
we are really interested in that topic.
Yeah.
So even
Mhm,
we are
mhm,
having Dr. Irshad Hussain with us. He is also
mhm.
working in the computational biology. He has done his PhD from NIT Silchar and working as an assistant professor in Shiksha Anusandhan,
Yeah.
Bhubaneswar.
Yeah.
So he is also in the same domain, and sir is also taking a session in this FDP later on.
Yeah. Yeah.
So
Welcome,
I
Dr.
hope,
Irshad. Yeah. Yeah, this is nice. This is nice. Computational biology is very attractive one, and
yes,
uh,
sir.
yeah,
I
the
actually
only thing
first
is,
day,
yeah,
yes,
yeah,
sir,
yes,
yes, sir.
please, yes, please, yes, please.
Sir, actually, I first met with Dr. Isad Hussain, and then I discussed about this SNN in 2022. And
Mhm,
then onward, I am just looking for the resources, and I am continuously eager to learn the thing. So
mhm.
intentionally, I have put him for the lecture. Then later on, I saw that you have selected this topic. So
No,
this topic is,
see, yeah. So actually, Dr. Surinder, I was talking about with him. He said, "Sir, I mean, if you could put it something that way, something related to more on this one, because now I'm working with the medical people here,
yes,
so
sir.
that give you more insight. But one of the scholar is working with me since last 3 years, and we have put some published few articles, and we have published very recent articles on conscious behavior and all those things there, and the results are very encouraging. The
Mhm.
work is more on AI side in that article, that how the consciousness concept can be added to our spiky neural networks,
Oh,
and
great,
yeah,
sir.
so
So
you can
definitely, I
check.
will connect with you, sir, for this one.
You always welcome anybody who's interested. Always welcome. We are
I'm very much
open.
interested in, yes, sir. I'm very much interested in working in this computational biology. But
Yeah.
my area is the cyber security, so I try
Mhm,
to map it on that side. But
mhm.
definitely, your guidance will be very much helpful for us also.
You are always welcome, Prof, along with anybody who's interested in the recent article. I think I can share the link with you. If you are interested,
Yes,
you
sure,
can
sure, sir.
go you can go through it. Uh,
Definitely.
just give me a second. So I'm sharing the ar archive link. So from there you can download it. Okay.
Sure, sir. Sure, sir. Thank you so much, sir. So, participants, I hope the session was very much insightful, and even I was very amazed. Means the way sir has explained, it was in completely a sequence. So, uh, thank you so much, sir, for your time and the time you have put on building these slides and this, preparing for the session.
Yeah, most of the time the scholars they helped me a lot in preparing these slides.
Yes,
Otherwise,
but
I
anyway,
I
yeah,
wish
yes,
to
sir.
add many more, but yeah, due to a bit short of time not able to add that much stuff there. But yeah, this one is even okay. I'm sharing few things here. Uh, uh, just give me a one more second. I'll send the link, and then this one is web page. You'll find more details on this my web page. My mail ID and details are there. If anybody is interested, he or she can put write a mail to me very far. I'll try to respond as quick as possible. So they are welcome, and very soon I'll update this website with all the PPTs. So you earlier PPTs are already there. Anybody who is interested can download the codes and PPTs and all these things from the website. These PPT, after some modifications, corrections, I'll upload it so that you can also use most of part of it. And if anyone has any question, you can mail me either at MNI, these IDs I can put it, MNIT mail ID also. This one is my MNIT mail ID, and you can mail me on use your mail ID also. So you can mail either of these mail IDs, you can put your questions. Yeah, thank you.
Thank you so much, sir.
Okay, Dr. Vikas, thank you. I think there is no many queries, nothing,
Yes.
no any discussions required.
No more queries, sir. Yes, sir.
Yeah.
Thank you so much,
Okay,
sir.
and I like to hear if I get it. I mean, it's like some time. I'll try to find a time to hear Dr. Ishal Hussain also. Maybe some
Sure,
new
sir.
insights will come there. Thank you very much.
Definitely.
Thank you.
Okay. Thank you so much, sir.
Thank you, sir.
So, participants, so participants will be joining back the same time for the session. Thank you, sir. Thank you, Surendra sir. Thank you, Asutosh, and thank you all the participants.
Thank you, sir. Thank you.
So we are
Thank you.
closed for the day. We are closed for the day.
Okay, sir.